So hello everyone, welcome back to my channel. So in the last videos we just started discussing about the tools of recombinant DNA technology. So I hope you remember the topic. Okay, we have completed the first tool. So what was the first tool? Yes, it is actually the restriction endonucleases. So we have already completed the first tool, restriction endonucleases, and we were discussed about the action of the enzyme restriction endonucleases. Okay, so that also we were discussed in the last class. Now we are moving on to the next tools of recombinant DNA technology. So the tools what we are going to discuss is actually DNA ligases. So already along with the restriction endonucleases, we were already discussed about the DNA ligases. So DNA ligases which are used to join the DNA fragment. I told you that here actually after the action of restriction endonucleases, we will get the fragment. Now the fragment we have to join with the vector DNA. So there we are using DNA ligases. Okay. So DNA ligases, it will make a like a phosphate bond. So we can say that a phosphodiester bond. So DNA ligases will make a phosphodiester bond in between the nucleotides. Okay, so that you have to keep in your mind. DNA ligases will make a phosphodiester bond between the nucleotides. Okay, so that's all about DNA ligases. And next, DNA polymerase enzyme. So here DNA polymerase enzyme which are used for making strands of DNA. Okay, so strands for making new strands of DNA, we have to use a DNA polymerase enzyme. Now the next tool that is very important, somewhat detailedly we have to study, that is actually the cloning vector. Okay, so cloning vector. So what is actually a vector? So why we are using a vector? See here, a vector means it is used to carry the foreign substances. As we have already studied that DNA fragment. See, in a DNA, we are incorporating a small fragment. Just imagine that this is the DNA. So in this DNA, we are incorporating the small DNA fragment. See, as like this, and we will make a recombinant DNA. We got it. Now just imagine that, see, for carrying this small piece of DNA, simply if we insert the DNA into the host organisms, the host organism will not accept it. So what we have to do it, we should have to make a vector, we should have to make a carrier. So this carrier, it will transmit the DNA, the small piece of DNA or the foreign DNA. So for carrying the small piece of DNA, we are using actually a cloning vector. Now see here, generally we can say that there are many cloning vectors are there. Okay, bacteriophages can also have a cloning vector, and agrobacterium tumefacients, plasmids, that and all actually we can use as a cloning vector. Now here we are moving on to the portions like an artificial vector. Okay, so we made an artificial vector. So an artificial vector means a vector which is having all the desired character which are required for us. Okay, that is actually artificial vector. So this is actually the artificial cloning vector. So we will call it as PBR322. So PBR322, it is actually a plasmid. Okay, it is an artificial vector discovered by the scientists Bolivar and Rodriguez. So P is actually the plasmid, PBR, Bolivar Rodriguez. Okay, plasmids, Bolivar Rodriguez 322 is actually the number in which this enzyme is isolated from the laboratory. Okay, that is the order. Okay, now in this PBR 322, we have to study the characteristics of this PBR 322. First, we can start with ORI. ORI means actually the origin of replication. Okay, origin of replication. So that is actually ORI, origin of replication. Now, so what is actually this origin of replication, ORI? or what is the use of this ORI in the vector? Actually, as we know that a vector, actually generally each DNA is having ORI site. So it is required for the replication process. See here itself I have mentioned origin of replication. That is, it is the region where the replication start or replication initiate. Okay, so ORI site, it is required for initiating a replication. Okay, so that is actually ORI site. Okay, now here, so when any piece of DNA is actually ligate into the site of ORI, it will replicate in the host organisms. Okay, that you keep in your mind. Now the next important character in the ORI site, actually it is required for controlling the copy number. Okay, so it is the region where replication initiate, as well as it is the region where the, or it will control or regulate the copy number. Okay, so here see, just for example, if you want to get more copies of the small DNA fragment, so we should have to select a vector which is having high copy number, so we will get more copy of the desired DNA fragment. Okay, so that's all about actually the ORI. Now see, you can look at here the next character of this PBR 322, that is actually the selectable marker. Okay, so the first character, that is actually ORI. Second one is actually the selectable marker. So selectable marker. See here, marker. What is the use of this marker? For identification, marking, right? So the term itself indicating selectable marker. So if we want to identify the transformant from the non-transformant, okay, or we can say that if we want to identify the recombinant from the non-recombinant, we have to use the selectable markers. Okay, so normally the antibiotic resistant genes such as like we can say that kanamycin, chloramphenicol, tetracycline. So this act as a selectable marker. So in normal plasmids, this selectable markers are actually absent. Okay, so here we can say that two selectable markers are there: antibiotic resistant gene, tetracycline resistant gene. Actually, it is a, I already told the synthetic artificial vector. That is why it is having two selectable marker. Now, in a gene, lac-Z gene, which is code for beta-galactosidase, which is also act as a selectable marker. So that detailedly we can discuss in the upcoming portions. Okay. So that is the second one, selectable marker. Then next one, cloning site or recognition site. Okay, so cloning site, or we can say that recognition site. See, in cloning site or recognition site, see, each vector should possess minimum one cloning site. Actually, one cloning site it should be there. If it is more than one, actually it can cause a complication in the recombinant DNA technology. So here you can see that PST1, PUV1, EcoR1, CLAV1, HIND3, BAMH1, SAL1, PUV2. These are the recognition sites. Okay, so that also you have to keep in your mind. So recognition site or cloning site is also required in a vector. Okay, now next ROP. See, ROP actually it is a region which will synthesize actually the protein which are required for the replication process. Okay, so that also it is required. So these are the major characteristics of actually a cloning vector. Okay, so this is the characteristics of cloning vector PBR 322. So characters of PBR 322 that we have already completed. Now we are going to discuss about the selection process. As I have already told you that in PBR 322 we are using selectable markers. So by using a selectable marker, how we are separating the recombinant and the non-recombinant. So that we are going to discuss. So the first topic that is actually antibiotic resistance or selection by antibiotic resistance. So as I have already told you about the ampicillin resistant gene and the tetracycline resistant gene. Just imagine that a small piece of DNA. So this is actually the small piece of DNA. So a small piece of DNA we are ligating at the site of BAM H1 of the PBR 322. Okay, so BAM H1 site of the tetracycline resistant gene in the PBR 322. So what will happen when we will ligate the new fragment into the vector DNA? So what will happen when we will ligate at the BAM H1 site? So just imagine that we have ligated. Now this is actually the sequence. Okay, just consider this as actually the new sequence. Okay, so here we ligated. Okay, so when we ligate a small DNA fragment into the BAM H1 site of tetracycline resistant gene in the PBR 322, see, they will lose the tetracycline resistance. Okay, so the recombinant, now this is called the recombinant. So the recombinant will lose the tetracycline resistance. Just imagine that this is actually the non-recombinant vector, a vector which is having all the characters. The non-recombinant vector, it is actually having ampicillin resistant gene, tetracycline resistant gene, and all the other characters are there. Just consider this. All the other character like ORI site, ROP is here. So all the other characters are there. Every characters are there. So this is actually a non-recombinant. This is the recombinant. Okay, so we have a more non-recombinant and a recombinant. Now the main aim is actually we have to select the recombinant from the non-recombinant. So there we are using the selectable markers. Okay, now actually I will tell you how selectable markers are used here. So first we have to take all the DNAs, either recombinant and non-recombinant, and we have to place the DNAs or we have to place the vectors in the first we have to place in the ampicillin medium. Okay, ampicillin medium. So AMPR, ampicillin resistant gene is present here. So first we have to place in the ampicillin medium. Just imagine this is actually the ampicillin medium. So when we will keep both recombinant and non-recombinant in ampicillin region, sorry, ampicillin medium, what will happen? See, ampicillin is actually antibiotic. So here it is having antibiotic resistant gene. Is it not? Both non-recombinant and recombinant is having ampicillin resistant gene. Okay, so when we are adding both non-recombinant and recombinant in the ampicillin medium, see, both will resist the ampicillin. Okay, both resist ampicillin. Now after this we have to take another media which is actually having tetracycline, which is having actually tetracycline. So when we will keep both recombinant and non-recombinant in the tetracycline. So first we have to place in ampicillin. So from there we have to keep both recombinant and non-recombinant in the tetracycline. So what will happen? See here, recombinant, it doesn't have a tetracycline resistance. Okay, non-recombinant it is having tetracycline resistance. So here recombinant it will grow in the medium of ampicillin, not in the medium of tetracycline. But non-recombinant it will grow in the medium of ampicillin as well as in the tetracycline. So this is actually one of the mode of selection. Okay, now the next mode of selection is actually we can say that inactivation of antibiotic. So already I have told you about the LAC-Z gene. So LAC-Z gene it's code for beta galactosidase. Okay, so this selection process actually it is done by keeping the recombinant and non-recombinant in the a chromogenic substrate. So based on the color production in the chromogenic substrate, we can select the recombinant and non-recombinant. Okay. So as I have already told you about the first selection process, actually here this selection process is actually somewhat complicated. Why it is complicated? That is actually one of the demerit of PBR 322. That means first we have to place in ampicillin, then we have to place in the tetracycline. Okay, so that is actually a complicated process. Okay, so continuously we have to transfer from ampicillin into tetracycline. So in order to overcome this demerit of PBR 322, another cloning vector is made, that is actually we can say that PU8, that PUC8. Okay, so here University of California, they made a plasmid. Okay, so just imagine this is actually the plasmid. So here it is having ampicillin resistant gene and a LAC-Z gene, which is called for beta galactosidase. Okay, keep in your mind. Now just imagine a small piece of DNA we are actually incorporating in the like LAC-Z gene. So what will happen? So when we will ligate a small piece of DNA in the LAC-Z gene, okay, we can create a recombinant. So just imagine that this is non-recombinant. In the same way, this is actually the recombinant. Just consider this as a small DNA fragment that we ligated in the LAC-Z gene. Okay, so here we can see the AMPR, and this is actually the ORI site. Okay, just imagine. So this is the non-recombinant and this is the recombinant. Now both we have to keep it in the chromogenic substrate. So when we will keep it in the chromogenic substrate, see the vector which is having LAC-Z gene, it will produce a blue color in the chromogenic substrate. Have you got it? So in the chromogenic substrate, the LAC-Z gene, it will activate and the enzyme produce a blue color. Okay, now in the recombinant, actually that LAC-Z gene is absent, so it do not produce any color. So we can say that a white color. Okay, so we will call it for this selection, we can say that blue and white selection. Okay, so this selection, it is actually also called inactivation of antibiotic. So when we are ligating a small foreign DNA into the LAC-Z gene coding for beta galactosidase, actually it will lose the enzyme will not be activated and the color will not be produced. If the vector which is having Z gene, LAC-Z gene, it will produce a blue color in the chromogenic substrate. So when we will talk about the examples of vectors, so plasmid act as an example of vector. Plasmids are actually the circular extrachromosomal autonomously replicating DNA. So plasmid can act as a vector. Then bacteriophage, it is also can act as vector. Then yeast artificial chromosome, bacterial artificial chromosome, phagamid, cosmids and transposon. So these are the examples of vector. Now, when we will talk about the vectors for cloning genes in plants and animal, see here, there is a pathogen which is called Agrobacterium tumefaciens. So these Agrobacterium tumefaciens, it is actually a pathogen for several dicot plants. Now this Agrobacterium tumefaciens, it will deliver a piece of DNA known as tDNA into the dicot plant. So this tDNA, it will transfer normal cell into cancerous cell. Okay, so Agrobacterium tumefaciens, the DNA, it can transfer normal cell into cancerous cell. Now, see this cancerous cell, it will make a chemical which are required for the pathogen. So that is a major purpose of this transformation. Okay. Now, these Agrobacterium tumefaciens, we can use as a vector. So for cloning genes in plants. In case of Agrobacterium tumefaciens, we have to insert the DNA, the desired DNA into Agrobacterium tumefaciens. So when we will insert the desired DNA, it will be non-pathogenic. Now this DNA of Agrobacterium tumefaciens, it will infect the plant cell, so we can deliver the like a desired gene into the plant cell. So that is actually in case of vectors for cloning genes in plant. Now, in the same, vectors for cloning or vectors are used to clone genes in animal. See, as like agrobacterium tumefaciens, here also retroviruses. It can also causes cancers to the animal cell. So here also the modified retroviruses we can use for transferring the gene into animals. Okay. Now, see, there is one more terminology that we have to keep in our mind, that is actually shuttle vector. So, shuttle vector means the vector which can grow or which can replicate both in eukaryotes and prokaryote. That means it is having a ORI site for replicating in plant cell and sorry in case of eukaryotic cell as well as in case of prokaryotic cell. So here the examples of shuttle vector is actually YEP, that means yeast episomal plasmid. Then the another example is actually modified TI plasmid. So yeast episomal plasmid and modified TI plasmid act as a shuttle vector.